welcome back. Digging deeper into talking about what does it look like at the genetic level when we have a new mutation appear that produces a new allele that's beneficial, and then what does it look like when the environment changes, and then we see relative fitness change due to that change in environment. So the relative fitness of an already present allele that's already present in the population. So what does this look like? So this image, it's so little, I don't know if you can see. So what I've put up at the top is just to remind you what we're looking at is a stretch of a chromosome. So all the little AATs, oh, it went away. Oh goodness gracious. Y'all can't see that. Come on. That's really annoying. There we go. Okay, so at the top are all the A's, T's, C's, and G's, just to remind you that what we're looking at is a stretch, a piece of a chromosome. And then what each of the little blue squares are, so these are all blue squares. What those are is those are representing neutral mutations that exist in this population. And so I'm just showing you, what is this? 1, 2, 3, 4, 5, 6 stretches of this same chromosome. And each of those little blue squares are representative of neutral mutations. So what does that mean? It means that these mutations are not affecting the fitness of this population. So that could mean that there are synonymous mutations. It could mean that there are non-synonymous mutations that don't affect fitness. So whatever's going on, those mutations are not affecting the fitness of this population, the ones that are in that, the blue color. And then let's imagine that this yellow, this yellow is a new mutation. It's a new beneficial mutation. A new beneficial mutation, okay, and it has a high relative fitness. So if it has a high relative fitness, that's our W, that means that there's strong selection. Strong selection, so that would be our S, our selection coefficient. So we have a large selection coefficient in this case. So what's going to happen is that very quickly, remember, in very few generations, the frequency, the P, of this allele is going to increase very rapidly. So we have a big delta P. So we have this is our imaginary population. What we're seeing is neutral mutations, and then we have a new beneficial mutation as indicated by yellow. So as this population reproduces, what we're going to see is that this individual who has this new beneficial mutation that has a high relative fitness, producing a large S, so a large selection coefficient, we're going to have a big delta P. So a big change in frequency from one generation to the next. So essentially, this individual passes on this allele at a higher frequency then do all the other individuals pass along their alleles. And so what you're seeing is, because this happens very rapidly, is that because it happens in short evolutionary time, what would be a good color for this? That this chromosome gets passed on intact. So it gets passed on to subsequent generations with the same order, the same alleles, in the same order, in the same places. So what does that mean? So that means that these alleles that are close, these mutations, these neutral mutations that are close to the beneficial mutation, get carried along with it. Why does that happen, and what is that called? So if we think about these places on the chromosome, right, if we're going to go through crossing over and recombination, remember that crossing over and recombination is unlikely to occur in places that are close to each other. It's less likely to occur in places that are close to each other. So if this new mutation is here and it's between these two neutral mutations, in short evolutionary time, a crossing over event is less likely to occur to separate those from each other, because there are fewer crossing over events, right? So if this is from one generation to the next generation, we've got one chance, or two chances, or three chances, depending on how many offspring we have, to cross over and recombine in that location. Okay? So remember that linkage disequilibrium is when we see alleles inherited together more likely than we would expect by chance. And usually this is because these genes are physically close to each other on a chromosome. So if we have a new beneficial mutation evolve, and it's very beneficial, and we have rapid evolution, then the alleles, the allelic variations that are close to it on the chromosome, get carried with it forward in evolutionary time. Okay, so this, we call this hitchhiking. So these alleles that are not beneficial are hitchhiking with the beneficial allele. And the reason that they're able to do that is because this happens, the increased frequency of this allele happens so quickly in so few generations that crossing over and recombination is very unlikely to happen between those alleles. Okay. It might happen occasionally, but it won't happen often. So what you'll get over time is fixation of that section of the chromosome in the population. So you'll see alleles that were not beneficial, that are close to the beneficial allele, get carried forward into subsequent generations. And as this beneficial allele spreads and goes to what we call, remember, fixation, so as it spreads and it goes to a frequency of 1, which we call fixation, the alleles that are next to it, just physically next to it on the chromosome, are also going to go to fixation. Okay? Boom. They also go to fixation. Just because they're physically close to it, because they are in linkage disequilibrium. Okay, so what does this mean? What does this mean? This means that a non-beneficial allele can spread by natural selection. So you can get an allele that's not beneficial, you can get an allele that's neutral, and it will spread by natural selection in the case of this rapid spread due to a new mutation that has a high relative fitness and strong selection spreading rapidly through the population. Okay, so what this is called a hard selective sweep. And that's when you have a new mutation. Okay? New mutation is when we see this. And the key to this happening, to this hitchhiking, is that this happens rapidly. So we get a very rapid rise to fixation in very few generations. And so that prevents crossing over and recombination from happening often, or maybe at all, in this particular area of the chromosome. How cool is that? So this is actually a technique that we use, like for real, in real populations to figure out what's going on. Was this a new mutation? Is this why do we have this section of the chromosome that's fixed? Okay? So if you see that whole section of that chromosome, it's all fixed, all the same alleles. We had a hard selective sweep. Okay, but what happens when we don't have it, when it's not a new mutation? So we talked about we can get a new mutation, but then the other option is that we can have an environmental change. Okay, so in this image, I've just changed the color, is all I've done, to kind of indicate we're talking about something different. So in this case, even the little rectangle that's a different color, it's a neutral mutation at this point. These are all neutral mutations. So because remember, we're not getting a new mutation that's beneficial, that's adaptive. The environment's going to change. So everything, even the sort of pinkish purple allele that I indicated with the arrow, they're all neutral at this point. They're not beneficial, they're not deleterious. They're just there. Okay? So they're just there in the genome. And so what that means is that over the course of long evolutionary time, this mutation that we're interested in, this allele, okay, this different allele, it's not beneficial, it's not deleterious. So there's a lot of time for evolution to happen. There's a lot of time for crossing over and recombination to happen. And so what that means is that over evolutionary time, there was lots of opportunity for crossing over and recombination to break up that physical association. Lots of opportunity because there's so many generations for crossing over and recombination to occasionally happen in that place. So we get a variation in which other neutral alleles this allele is associated with. So in this top, this allele that we're interested in is not associated with either of the two alleles it was originally associated with. In this one, it is still associated with the original two neutral alleles it was associated with. Here we have a different situation, okay? Because we had lots of time. Because nothing was selecting for this allele. Okay. Then imagine, boom, we have an environmental change. Okay. We have an environmental change. And so now we're getting selection for this allele. In this case, this is a soft sweep. And in the case of a soft selective sweep, rather than seeing the alleles that are nearby on the chromosome carried with the beneficial allele, what you'll see is fixation only at that spot, only at that location, because there's variation. So what you're going to see then over time, I didn't do this. Hang on. Over time is rather than seeing fixation of a whole section of the chromosome, you're just going to see fixation of that one allele and maybe just a little bit of flanking sequence. So that one allele and maybe a little bit of flanking sequence, it's going to be identical. But all those other neutral mutations, they're going to look different in different members of the population. Because crossing over and recombination had already broken up those associations long before the environment changed and that allele suddenly became beneficial. So in a soft sweep, you're only going to see a very narrow part of the sequence that gets fixed. Only going to see a very narrow part of the sequence that is going to be identical for all members of the population. So that's how the genome changes and how we can identify whether we had a new mutation that appeared and suddenly swept through the population in the case of a hard sweep, or natural selection acting on standing genetic variation in the case of a soft sweep.